Hi everyone, Jack Cush here at Room Now. We're in the booth. I'm joined by one of my mentors, one of the people I looked up to for so many years in science and medicine, Dr. Charles Dinarello. Charlie is now at the University of Colorado in Denver, has an illustrious career. His CV is measured in pounds, not in publications. It's so darn long. He told me earlier you just got your one NIH grant renewed that was originally given to you in what year?
1978.
And it's been renewed again here in 2022. Unbelievable. This man has a career unmatched in science, responsible for all we know about interleukin-1 and so much else in the inflammatory disorders. I saw him and he told me his story. I think it was almost 30 years ago when he knew I was a young guy interested in anakin or an IL-1 or whatever, that how to treat recurrent pericarditis with an IL-1 inhibitor, IV and high dose. I thought, oh my God, this is either brilliant or crazy. Of course, it was brilliant. So, Charlie, always great to see you.
I'm so glad to see you. I haven't seen you in a long time.
Too long. So tell our audience, how did you stumble? Did you stumble onto IL-1 or what happened?
No, it's actually a wonderful story. I was a second-year medical student at Yale Medical School, and Yale was a unique medical school because they required you to write a thesis for the MD. It's the only medical school in the United States that actually requires a thesis for the MD degree. So I started in a wonderful laboratory that was headed by Elisha Atkins and Phyllis Bodell, and they taught me how to do the fever assay, which is essentially putting a rectal probe in the ass of a rabbit and injecting supernatants from white blood cells intravenously, and that would produce a very dramatic response. It was so dramatic it still stays with me today. You'd inject the supernatants from white blood cells, could be human white blood cells, and you would see the ears of the rabbit get ice cold as the blood shunted to internally, and then the fever would just go sky high. In 50 minutes it reached a peak, and then suddenly the ears got really warm and the fever came down. It was such a dramatic thing. I was 22, 23, and I promised myself I was going to figure out this fever molecule. Well, that fever molecule now is called interleukin-1 beta. I wear this hat. I'm proud of it. I'm not ashamed to say I'm proud of what we did. But the story is really a long story. And it starts with something that was very difficult. Purifying a molecule from human white blood cells that was present in incredibly small amounts. But after, I would say, 5 to 6 years, and it was a lot of disappointments, we finally purified in 1977.
Wow.
We said at that time that this molecule was so potent it would cause fever at 10 nanograms per kilogram in a rabbit.
Right.
Nobody believed me. They all said, oh, he's got contaminated endotoxin. Everybody said everything was contaminated with endotoxin in those days. Today, interleukin-1 beta causes fever in humans at 10 nanograms per kilogram. You can even go down to 1 nanogram per kilogram. And perhaps the most potent cytokine. Now, along all of this story, no one had gene information. There were no genes cloned. The first gene clone was growth hormone, 1981. Genentech. Genentech started with the cloning of growth hormone. I said, why don't we clone IL-1 beta? It's not an easy problem. There was no PCRs in those days. No PCR. You're going to clone a molecule with no PCR technology. We know today it's really easy. It wasn't easy then. But we did it. I went to MIT. I was lucky because the subway from Tufts University, where it was, to MIT was only 2 stops on the Red Line in Boston. So I lived in the subway. We succeeded. We published the cloning of IL-1 beta in 1984. That was perhaps the real milestone in cytokine biology. What happened next was a miracle. You had the gene, you can make recombinant proteins, you can make an antibody, you can do all kinds of things. That exploded cytokine biology. At the same time, 1984, another molecule was cloned by Genentech called TNF. Now there were 2 of them. Obviously, I focused on IL-1, but I really respect TNF. It was a really interesting molecule.
Yeah.
mean, that's what this going on today.
Yeah. Much of this meeting is still
Yeah, I know.
focused on those two molecules.
Who was the hero of TNF? Incredibly interesting guy, a wonderful guy named Anthony Cerami. Cerami, I'm 100% Italian. He's only half Italian. We're still good friends. He discovered TNF. And he was a guy that said, if you make an antibody against TNF, you can cure rheumatoid arthritis. He has a patent on that. This guy was really a pioneer. I'm so happy that we're still close friends and we see each other often. But that was how cytokine biology was. There was IL-1 and TNF. Well, TNF took off because it's so important in autoimmune diseases. No one really paid attention to IL-1 until we realized that it was dominant cytokine in diseases that were not autoimmune, but they were called autoinflammatory. And the list of autoinflammatory diseases is huge. Not only does gout, that's what I spoke a couple of hours ago, was the importance of IL-1 beta in gout. TNF is not important in gout. But also, IL-1 is important in heart failure, diabetes, a variety of things that are autoinflammatory. And now we look at IL-1 beta blockade in cancer. And there you can really separate IL-1 from TNF. It's kind of a beautiful story of two important cytokines that sort of have a home in autoimmune diseases, and other one, IL-1 beta, a home in autoinflammatory diseases. And that's really the story. There's one other thing I want to talk about Tony Cerami that people don't know. When I watch television, and I do watch news programs, a lot, almost always. There's so many commercials, okay, of all these things, okay? And they all talk about, oh, I lowered my A1C, all these drugs lower my A1C. I get so many commercials on A1C. Do you know that Tony Cerami discovered A1C in the blood of his mother, who had type 2 diabetes?
The same guy who discovered TNF.
Yeah.
Pretty amazing.
He's an amazing guy. He's a nice guy. We're really good friends. He respects us for our work in IL-1, and I respect his work for TNF. I'm very lucky to live long enough to tell you the story.
The only way this could have been better is if Tony Cerami were sitting here wearing a TNF mask. That would have been an unbelievable interview. Tony, Charlie, great to see you. You're the best.
You're the best.
All right.
Thank you. Thank you very much.